All right, good afternoon, everybody. Welcome. This is the session on modeling methods here at the IDM Symposium. I guess the way to think about this session is that a lot of the work that you've seen presented here at the symposium is focused on a particular vertical, like a malaria, cholera, that sort of thing. And so here we'll be focusing the other way, cutting across the individual disease areas to look at some of the underlying methodology. And while we appreciate that these techniques are farther away from the impact and from the actual health decisions, I think it's really fundamental that we have this session in place because improvements like we'll see today across modeling methodology has the potential to lift up all of the analyses that we collectively do. So we got an ambitious schedule today where there's 5 talks in these 90 minutes. So we'll be aiming for maybe 13 to 15 minutes. Hope we'll have time for a question or two at the end of each talk. I'll be keeping time from the front here, and we'll have a diversity of different methods presented. So looking across kind of differential equation-based models, a good deep dive into some model calibration, and then we'll finish looking at some noise in these models. So without further ado, I'd like to introduce Vivek, who is joining us from Johns Hopkins University. Over to you. Thanks so much.
All right. Good afternoon, everyone. Nice to meet all of you. So my name is Vivek Murali. I'm a second-year PhD student at Johns Hopkins. And today I'm going to be talking about modeling non-exponential time distributions, primarily in compartment models. So let's just start with the most basic compartment model in epidemiology, which is the SIR model. And this is a very simple model characterized by 3 differential equations here. And such models like this are obviously very useful to study all kinds of diseases. It gives us, it's analytically tractable as well. We can do simulations very easy. But there's one fundamental assumption that we make when we make these sets of equations over here. So when we make these deterministic equations, what these equations really are is they represent an average or a mean of a lot of stochastic processes happening at very large numbers, right? So these differential equations here don't exactly care about how much time a given individual spends, let's say in the I state going to the R. It's just on average, an individual will move, at this rate. But if we take a stochastic analog of this and we stick to the fact that this is a constant rate, then the assumption that we're making here is that the waiting time in that I state would be exponentially distributed, as seen by this red line over here. And that's usually very mathematically convenient, but problem is it's not really realistic. There's just some data on serial interval period here that we could probably use with some assumptions, obviously, to characterize the I waiting time here. All right. So given the fact that the underlying distribution may not necessarily exactly be exponential, what can we do? So there are multiple ways around this. You could formulate integral differential equations around this as well to really get the correct shape of this. But one fairly simpler way is this thing called the linear chain trick, which is basically dividing this substate at I into multiple different K substates. And so, but when you divide them into multiple K substates, the rate through each state is significantly it's K times faster. So essentially intuitively what that means is I'm still taking the same average time to go from I to R, right? But I'm going through all these substates. So the mean is kind of the same. But what I'm really changing here is the variance of that distribution, which would then sort of match the underlying distribution. So I'll just give you a more concrete example to highlight this. So, we have this one transition rate and we have an exponential distribution. We divide that into 2 states. We have this distribution changing over here. Same thing. You can make it into 5 states, 25 states, 75 states. I mean, the list goes on. And you can see the distribution, while keeping the mean constant, is giving you a more narrow and a more narrow distribution. And if you go all the way to infinity, you'll get a fixed time delay. Is essentially what's going on. And mathematically, and this is the only time I will show the equation here, but this is essentially a convolution of multiple exponential distributions, which give you an Erlang distribution, or otherwise known as the gamma distribution with a integer k parameter. So the main beauty of this is that just by changing k and lambda, I can get a whole host of biologically plausible distribution time periods. So the same thing applies to this. What I've just shown you was going from the I to the R state. Easily you can apply to any compartment in the SCR model. Even from the E state, it gives you the latent period distribution. So like now that I've shown you like all these distributions, like one question we have is like just changing, like do different waiting time distributions really affect your model outcomes, right? So I'm going to show you this through just two examples. So let's for now consider an SEIR model, and we have M substates in the E and N substates in the I. So this paper by Waring, they did some literature search, and they actually found that there were some common diseases here like measles, hand, foot, mouth disease that had sort of the number of substates fitted to them. Exactly how these numbers were gotten was not so clear in literature, but the main point of showing you this is to show you that there are diseases that have this waiting time distribution periods that should be considered. And one of the most key figures in their paper is this. So let me walk you through this. So we have this 3D figure. The X and the Y axis are just the number of substates in E and the I state. So what they're doing is observing the estimated R0 value from varying assumptions of how many substates you have. And you have this huge surface over here. If we were to assume that E and I were purely exponential distributed, we would be all the R0 will be falling on this grid over here. And if we assumed a fixed time delay of some value, it will be there. And so the main point I'm trying to make here is that when we assume exponentially distributed infectious periods, we can get different values of just like your R0 from this estimation. There's another example here. Basically also an SEIR model with two additional states here. These two states represent just more of like control measures, like quarantine state and isolated state. So what this paper was doing, paper by Feng, what this paper was trying to do is evaluate how control measures, like how do they change if you adopt different assumptions of waiting time distributions. So when they assume that E and I are exponentially distributed, this was their result. Let me walk you through this first. On the y-axis is your cumulative infection number. On your x-axis, this is a little bit tricky because there are like two parameters here, but essentially what they're doing is they're varying one of the control rate parameters while keeping one of them constant. So you have two of these curves, and you're just like seeing how they vary with like relative to like each other. So the main point I'm trying to make here is that if you just draw a line, if you just draw a random line on the x-axis, there's not much difference. You'll probably say like the rate of being quarantined, the rate of being isolated, they both have an equal impact on your total infections. However, when they tested it on like three substates meaning when they changed the E and the I to like three substates each, and they ran the same simulation, there was actually a slight discrepancy here, telling you that one control measure might be better than the other in terms of bringing down your infections. And so like their main point here is then assuming different distributions would also give you conflicting assessments on what kind of intervention is actually good, right? And this example here was just like three states. This difference might be a lot bigger if I go to 5, 7, 8, or 9, which then leads us to this other question here, which is, given the disease, exactly how many substates do we choose? And what's the most appropriate number of substates we choose for a given model, right? And so that's kind of the overall goal of this project. It's to develop a tool that will estimate the optimal number of substates or parameters of our Erlang distribution from experimental data on waiting times. So just a quick overview of what the tool will look like. So we have some relevant data on waiting times. We do a maximum likelihood to find the best K and the best lambda, and the best K basically informs the user of relevant number of substates to be used in the modeling. So I'll walk you through this just through a very simple example here. So we have some data published in this paper from COVID-19. They basically have a histogram of incubation periods here. They were also kind enough to share the data exactly on the incubation periods on their link. So we just took that data, and slight caveat here, like we're gonna assume that for this toy example over here, that this incubation period could be used to model like the waiting time in E. Although that's clearly not very true because symptoms don't really correlate with being infected. So then we have the data here. So one thing is, if we went ahead and took the data and assumed an exponential distribution, what we would be doing is just taking the average of all that data, and that would kind of be the distribution that we will be assuming when we model just an SEIR. If, however, we do our maximum likelihood, we put the data through, we'll get, okay, that gave me a K equals to 5 and a certain rate. And how that will be translated in terms of modeling would be your E now has like 5 different substates that you go through. And that would be sort of your revised SEIR model. with whole lots of differential equations here that you could also use to do all your analysis, and would give you like a whole different simulation. Although in this Tau example, it's not that different. So if you look at it, if I just assume an exponential and just ran a Tau simulation here, this would be my curves. And if I did a Erlang goes to 5, I would have a slight shift of all my sort of curves. But this again is like a very Tau example. Like I'm sure if you model much more complicated states, the exact implications of what your results are will probably change given your underlying assumption. So that was essentially it. And this idea of using multiple states can apply to multiple fields. Okay, so one of them is even applying it to like a cell cycle phases. So you have like a normal cell cycle has like 4 different stages, like G1, S, G2, and M. So this paper also did something similar. They did a maximum likelihood and they fitted each of the waiting times to an Erlang and they got different numbers of states. But when we saw this, what I guess like caught our eye was the fact that sometimes you do get this really high number of states, like k equals 43, which is like, I mean, you don't want to be putting like 43 different equations in your whole model just to capture an accurate waiting time. So then another question we had was like, how low can we go on the number of k while still kind of capturing that time distribution, right? So that was kind of the second mid goal that we were like working towards. So just as a quick example, from just now, we took the data from the model and we fit it to k equals 43, and we also managed to code out something where we could get the lowest number of substates to be 32. I don't know if I have time to like really go through the like how you get the lowest substates in depth, but maybe I can just like talk it through right now. So let's say you have a model, okay, or your model of 32 states. What we did was just did a simple benchmarking. So we basically took that model, we drew some data from the model, and then we calculate the likelihood of that data given the model. You can do this 10,000 times and you have like a distribution of likelihoods that you can generate. And then what you do is you compare the likelihood of the empirical data given the model to the distribution, and you get a p-value to figure out how likely it is the data came from the model. That was kind of a whole mouthful, but I'm more than glad to talk you through that right after this. So that was our second like mini goal for this. And our third goal was we were looking at the Erlang distribution and wondering, okay, like that's great. We can do multiple distributions, but why are we constrained to these rates being constant? And that's one of the definitions of an Erlang distribution. Like you have the constant rates between these states. Like why can't these rates be maybe non-constant or not the same with each other? And maybe that would give a bit more flexibility in terms of modeling. So just to show you a little bit of what that looks like. So we have an exponential time distribution with that rate. And then if I fit in Erlang, I have like 5 states, for example, with 5 states with the same mean. And now if I were to vary the rates, I could still get different probability distribution shapes just from trying out different rates. And this could probably give us more flexibility in terms of capturing underlying distributions while still keeping the model overall mathematically simplistic. So that was quite a lot to go through at once, but that was essentially most of the work that we're doing now. These are all future works in progress, multiplication and so on and so forth. But the main point of this whole project is to create a tool that we will